How do Parkinson's biomarkers reveal brain changes? Imagine being able to see inside the brain and spot early signs of Parkinson's disease before symptoms even appear. That's what biomarkers are helping us do today. These biological clues can tell us about the underlying changes happening in the brain related to Parkinson's. They work by detecting specific molecules, cellular processes, and structural alterations that happen as the disease develops. Researchers are constantly discovering new biomarkers through studies and advanced technology, aiming to improve early diagnosis and treatment. One of the most important biomarkers is abnormal alpha-synuclein, a protein that tends to clump together in the brains of most people with Parkinson's. These clumps form structures called Lewy bodies, which are a hallmark of the disease. Scientists have developed a test called the alpha-synuclein seeding amplification assay that can detect these abnormal proteins in cerebrospinal fluid, which surrounds the brain and spinal cord. This test can identify Parkinson's-related changes with about 93% accuracy, even in people who have no symptoms yet. This means we might be able to catch the disease early, before motor problems or other symptoms start. Another way biomarkers reveal brain changes is through neurochemical measurements. In Parkinson's, the loss of dopamine-producing neurons in a part of the brain called the substantia nigra leads to movement difficulties. By measuring dopamine and its breakdown products in cerebrospinal fluid or blood, scientists can see the neurochemical shifts that happen as the disease progresses. Low levels of these chemicals can signal early or ongoing neuron loss, helping distinguish Parkinson's from other neurodegenerative conditions. Beyond proteins and chemicals, researchers also look at cellular and molecular changes. Parkinson's disease involves mitochondrial dysfunction, oxidative stress, and problems with how cells break down and recycle proteins. These processes contribute to neuron death. Biomarkers related to mitochondrial health, inflammation, and cellular stress are being studied to understand how the disease has advanced and how it might be slowed or halted. Imaging techniques are also vital in revealing brain changes. Magnetic resonance imaging scans can show structural brain differences, while positron emission tomography and single-photon emission, computed tomography scans, use special tracers to visualize dopamine activity. These scans can detect the loss of dopamine neurons in the substantia nigra and the connected areas of the brain. Importantly, these imaging biomarkers can sometimes identify changes before symptoms appear, offering a window for early intervention. Scientists are also exploring biomarkers related to brain connectivity and synaptic health. Changes in proteins involved in communication between neurons and alterations in brain networks can be linked to both motor and cognitive symptoms of Parkinson's. Although these are still under research, they hold promise for understanding how the disease affects brain function over time. In summary, Parkinson's biomarkers reveal brain changes by highlighting abnormal protein buildup, neurochemical deficits, cellular stress, and structural alterations. They are powerful tools for early diagnosis, tracking how the disease progresses, and testing new treatments. While much progress has been made, ongoing research continues to refine these biomarkers, with the hope of providing more personalized and effective care for those affected. Always remember, if you or someone you know is concerned about Parkinson's, consulting healthcare professionals is essential for accurate diagnosis and tailored management.